is an awful neurodegenerative disease that causes progressive loss of like motor function as well as neurological function. And it's caused by a trinucleotide repeat expansion. Don't worry, we'll get into this. But basically, it makes it so that in this Huntington protein, this important protein that helps regulate when things are made in the cell and where things hang out in the cell, in this protein, the instructions, there gets this typo made. And this typo was kind of like someone fell asleep at the keyboard while they were copying the instructions for this gene. And so you get lots and lots of the CAG. And the CAG, what it does is it kind of spells for the amino acid glutamine. So when the ribosome, the protein making machinery, goes to make this protein, it's going to see that long typo and it doesn't realize it's a typo, so it's just going to put in a bunch of glutamine. And this glutamine, this amino acid, is going to be really kind of like sticky for reasons that we'll talk about. It basically makes it so that now this protein can clump up and it can bring other proteins along with it and it can cause all these different problems. And it only takes a single bad copy of this in order to cause the disease. So it's what we call an autosomal dominant mutation. So let's talk about what all these different terms mean as well as the role of glutamine. So let's just start with some basic genetics. So the instructions for making different proteins, in this case the protein Huntington, are going to be written in the form of DNA. You can think of these original recipes, these genes, as kind of like recipes in a cookbook, where the cookbooks are chromosomes. So those are your whole collection of your DNA, is your genome. And then there's like 23 different of these chromosomes, pairs of chromosomes. And then the instructions, these original recipes, are copied into messenger RNA. So RNA is very similar to DNA, but it's a copy of it that you can actually take out of the nucleus. So out of this inner membrane compartment where the DNA is held, and then into the cytoplasm where the ribosomes or these protein making complexes are. And then these ribosomes are going to travel along those messenger RNA copies and piece together the amino acids or the protein letters in order to make the protein that the gene was telling it to make. Now, if the gene that was telling it to make the protein had like mutations in it or changes in the recipe, then you're going to possibly get changes in that protein. Sometimes these changes are good, sometimes they're not good. And when they're not good, they can cause disease, but they might not cause disease because, well, for every gene, you get one copy from each biological parent. And so for a lot of different genetic diseases, you actually need to inherit a faulty copy from each parent. We call these diseases autosomal recessive. And autosomal, this is just referring to that it's one of those like main chromosomes, and it's not one of the sex chromosomes. So for the XY chromosomes, things are a bit different because we only have like one copy of it, or two copies of the X, but I'm not going to go into that. We're talking about autosomal, so those are just like your main pairs of chromosomes. And in the case of a recessive mutation, this is basically where you have to inherit two bad or faulty copies of the gene in order for there to be a problem. So if you have one good copy, it's able to compensate. And a classic example I've talked about in the past is sickle cell anemia, where you have to inherit a faulty copy of the hemoglobin gene from each parent in order for this mutation to actually cause disease. But in the case of Huntington disease, what we have is the autosomal dominant disease. So just dominant means that you only need one bad copy in order for the person to have the disease. And so this can be caused by a couple different reasons. So one is haploinsufficiency. So basically one copy can't make enough to compensate for the loss of the other copy. And the other is dominant negative or gain of function. This is where the bad copy itself is toxic. So that's not just that you're missing the good copy, it's that you have like the bad copy is actually causing problems in the cells and that sort of thing. This can be, it's gained a new function that's making it do other things, or it could just be that it's not working. And so then it's kind of hogging the machinery that the good copies would need. And so in the case of Huntington's, it's largely thought to be gain of function, this dominant negative effect, where the bad copy is kind of clogging up cellular machinery. And it's even kind of causing these aggregations where it's including the good copies. So the bad copies are clumping up and they're kind of taking the good copies with them. And the bad copies are also, because they're causing all these problems and the proteins are clumping up and stuff, they're going to be taking away the machinery that the cell uses in order to fix the clumped up proteins and kind of clogging up all that machinery and causing problems. End result, a single copy is going to be able to cause disease. So we call it autosomal dominant. And in the case of Huntington's, this disease is caused by mutations in this protein called Huntington. And it's going to be a special type of mutation that we'll talk about called a trinucleotide repeat expansion. In this case, it's going to be a CAG repeat. So CAG is the RNA codon that's going to tell the ribosome, the protein making machinery, to put in this amino acid, this protein letter glutamine. And it's going to kind of tell it to put in a lot of glutamines. And so normally this Huntington protein, it has a stretch of glutamine, like from 6 to 35 glutamines. But in the case of Huntington's disease, you can get like 40 or more CAG repeats that inevitably leads to Huntington's disease if the patients live long enough. So the longer the tract that the patients inherit, the earlier the disease onset typically. Although there's a lot of different factors that can go into play, and scientists are kind of trying to figure out better what is actually kind of modifying or what makes it so that people are getting disease symptoms at different ages, despite having the same number of repeats. But in general, the longer the repeat length, the earlier the onset of the disease. And a lot of this is thought to be because this glutamine protein is able to kind of form, it's able to act really sticky and cause this protein to clump up and to bind to other machinery in the cells, hog machinery, sequester proteins, various things like that. And so we'll talk more about why this is. But basically for now, just think of this glutamine as being long and sticky. So it's really flexible. It's got this long chain. And at the end, it has this amide group. And for reasons I'll get into, this amide group is able to form these interactions with other molecules. It has partially positive and partially negative parts. And it's able to form these what we call hydrogen bonds. These hydrogen bonds are normally, we think of them as being in like the backbone of a protein because the backbone has amide bonds. These are what are linking individual proteins together, the individual amino acids together. But in the case of glutamine, you also have an amide bond in the side chain or in this unique part of the amino acid. And so you have this group sticking out that acts a bit like the backbone. And so it's able to form similar interactions. And these are the types of interactions that are crucial for maintaining protein function, like folding and things like this. And so if you can imagine that having these interactions coming from not the backbone, it can play important roles, which is why Huntington's protein, that normal CAG length is really, that normal CAG repeat is really important, allowing Huntington to form as a kind of like scaffold to bring different proteins together. Maybe it's not exactly sure all the things that it does, but some of the things are like regulate transcription, so regulate the making of regulate the making of the messenger RNA from the gene, as well as regulate other sort of signaling and things like this, because that CAG repeat is going to allow it to form as a scaffold. So having a few CAGs is good, but having a lot is going to be bad. And the CAG, remember, that's happening at the DNA level and the RNA level. And then this is causing the ribosome to put in this glutamine. And then this glutamine is going to be able to form a bunch of bonds. And then this is going to allow it to connect different molecules. But it's also, if you have too many of them, going to cause the protein to clump up. And there's also problems that can be caused by, like, at the level of the messenger RNA itself. So the CAG repeat, it can kind of cause the RNA to fold up and bind to different RNA binding proteins, and then kind of steal those proteins that other machinery needs as well. So there's a lot of research being done as well onto the problems that could be caused by the messenger RNA, in addition to the problems that are caused by the protein. So let's talk a little bit more about this mutation, because it's a kind of interesting sort of mutation. It's called a trinucleotide repeat expansion. So Huntington's disease isn't the only disease caused by this like trinucleotide repeat. So these trinucleotide repeats, depending on the trinucleotide, depending on that 3-letter code, that codon, it'll spell a different amino acid. Or if this repeat expansion is like before is in a regulatory part of a gene, it can just mess with the making of the gene like itself, like whether or not the gene is made, the protein is made, rather than making actual changes in the protein itself. And we'll talk about this in a minute. But there are also other diseases where you have this poly-Q repeat. So Q is the abbreviation for glutamine. And so in this case, you have a repeat an expansion in that in the DNA instructions where you have that CAG tract. And then this is telling it to put in more Qs, so more glutamine. And so other examples include spinocerebellar ataxias and spinal bulbar muscular atrophy. And so a lot of the research that is done on Huntington's can benefit these other diseases, as well as vice versa. Research on those diseases can benefit Huntington's research. So what are these? How do these repeat expansions form, and what do we know about them? So in order to kind of talk about this, I want to go back and talk about types of genetic mutations in general. So when we talk about the terminology, so the original instructions for making the proteins are going to be in the form of DNA, and then in transcription, we make that RNA copy, and then translation, we make a protein from it. So the DNA is going to be the permanent instructions. The RNA is just going to be like a copy that you can make different numbers of copies through regulating transcription, so that you can regulate how much of the protein is made in translation. And then when you don't want any more protein to be made, you can then degrade this RNA without actually having to mess up with the DNA. And if you want a lot of the protein, you can make a lot of RNA copies with only just having that original single copy of DNA, or two copies, because you have one from each parent. But so the changes, any permanent changes are going to have to happen at the level of the DNA. And so with the level of the DNA, this is where you actually get the mutations, where you actually get the changes in the DNA. So we call changes in DNA mutations. Mutations in the DNA, if they're in the protein coding part of a gene, so if they're in the part where they actually have the instructions for making the protein, then they can cause amino acid changes in the protein. So actual changes in the lettering of the protein, which amino acids it contains. They might lead to changes, but they might not. This is for a couple of reasons. So one is that the genetic code is degenerate. What this means is that multiple codons can spell the same amino acid. And so these codons are going to be what is instructing the ribosome what amino acid to add. So in this, the RNA is going, which is a copy of the DNA, remember, it's going to be read by the ribosomes in these kind of like 3-letter chunks, these codons. And so the ribosome is going to travel along the messenger RNA, and it's going to go codon by codon. And at each codon, a transfer RNA with the matching, or like the complementary sequence, we call the anticodon, it's going to come and it's going to bring the corresponding amino acid. And so in the case of CAG, you're going to have a tRNA that has the complementary sequence. It's going to come and it's going to bring a glutamine amino acid. And then the ribosome is going to travel along and it's going to add another glutamine and another glutamine and another glutamine until there are no more CAGs in that sequence and you go to a different amino acid. And so in the case of the Huntington's disease, in the normal version of the protein, you would have like 10 or so repeats. And so the ribosome would go along and it would see one CAG when it put in a glutamine, and see another and put in a glutamine, and do that 10 times, and then it would go into some other amino acid. But in the case of Huntington's disease, in the mutant version, you'd be going along and you'd have a ton of CAGs. And so this ribosome is going to be traveling along, and tRNA is just going to keep coming and bringing it the glutamine. It's going to put a bunch of glutamines in a row, and then you could have problems. So that's what a codon is. It's the like 3-letter word for bringing the protein, or bringing the amino acid. And so each amino acid is going to have at least one codon that spells for it. But there are 4 RNA letters and there are only 20 amino acids. And so this means that you're going to have multiple codons that can encode for basically the same amino acid. So in the case of glutamine, it can be spelled by a CAG or a CAA. In the case of Huntington's disease, it's caused by a CAG because that's what happens to be in the DNA, and therefore in the RNA copy of the DNA. But if there was a CAA there, then the ribosome would also bring a glutamine. And so if there was a mutation that changed, say, the CAG to a CAA, you wouldn't actually see any change at the protein level. And so we can refer to this as a synonymous mutation. And this would be an example of a point mutation, where you're just swapping a single letter. And so this would be a synonymous mutation, and so we sometimes think of these as a silent mutation if we can't see any difference. I'll note that not all synonymous mutations are silent because it could cause changes at the mRNA level, but that's too complicated to go into here. And we're not even talking about this type of mutation in the case of Huntington's disease. But this was an example of a point mutation where you're just changing a single letter. And so in this case, it was synonymous, but you can also have non-synonymous, where you can have a missense mutation, where one protein letter is swapped for another. This is the case with sickle cell anemia, where you have this one amino acid, a glutamate, which likes hanging out with water, swapped to another amino acid, a valine, which is water avoids, and this causes the protein to form these long aggregates that can kind of clog up blood vessels. More on that in my sickle cell anemia post. But I just wanted to bring it up here as an example of a non-synonymous mutation, in this case, a missense mutation. A non-sense mutation would cause one of these codons that says to put in an amino acid, change it to a stop codon. And so the tells the ribosome to stop making a protein, even though you're really in the middle of a protein. So those are point mutations. And in addition to point mutations, you can have insertion or deletions. So collectively, we call these indels. And these can have different effects depending on how many are lost or gained. So because the codons are in multiples of 3, if you have a mutation that is a multiple of 3, you're going to gain or lose amino acids, but you're not going to change the reading frame. And so when we talk about the reading frame, basically, the ribosome can start reading in 3 different ways because these codons are non-overlapping. It's going to read codon by codon by codon, like step by step by step. But it's not going to go, it's going to go 3 at a time, steps, not 1 at a time, steps. And so if you start reading in different places, you're going to get different things. And so if you have a mutation that changes by a multiple of 3, well, it's going to change the number of letters, but it's not going to change the actual, like the reading frame. But if you have a non-multiple of 3, then you get a frame shift and you get lots of letters changed. In the case of Huntington's disease, it's a trinucleotide repeat expansion. So 3. So we are changing by 3. We're just adding more CAGs. So we're not going to change the reading frame. We're not going to change the rest of the protein. We're only going to affect the part of the protein where we're going to be adding more of those glutamines in. And so this is an example of a trinucleotide repeat expansion. What happens with Huntington's disease is that you can actually get this expansion be worsening, and kind of like people can inherit more CAG copies than the parents had. And so this can make it so that even if the parent is healthy, even if the parent had a faulty version of it, had a healthy version of this Huntington's gene, they could still pass a bad version on to their child. Because what can happen is you can have this thing called like genetic anticipation, where basically the repeat expansion gets longer in the parents' germ cells, and then that gets passed on. And the reason why gets longer, there's a lot of research into this, but it's thought it has to do with kind of like during replication. So during the making of copies of this DNA for each of the new cells to have a copy, what happens is that the machinery can kind of slip up. And when it slips up, it could kind of lose track of where it was and add more copies in. It's also thought that DNA repair might play a role, so that when the cells basically when there's a break in the DNA and then the cells try to repair it, they might end up adding more of the CAG, so making the repeat worse. And then if this happens in those germ cells, it can get passed on. If it happens in the non-germ cells, so just like your normal cells in your body, not the sperm or the eggs, and what happens then is you can get what's called somatic repeat expansion. So that's not going to get passed on to offspring, but it would be passed on to any sort of daughter cells that arise from that cell. So there's a lot of research going into the role of somatic expansion in Huntington's disease. Since it's known that the longer the repeat, the earlier the disease onset typically, it's thought that if you have somatic expansion, this could cause a shorter repeat length to then reach the threshold where you're going to get problems. And this is an especially attractive hypothesis or theory because, patients typically don't get symptoms of the disease until they're in their 30s or to 50s or so. And so one possibility is that somatic repeat expansion is kind of causing the protein to kind of cross the threshold where if you have less than 40, you typically don't have symptoms. But then if you go over 40, you almost inevitably do. And so it could be that you're kind of having repeat expansion that's causing you to get over the threshold. So this was actually a research project I was looking into when I was an undergraduate, and I did summer research experience at Steve Finkbinder's lab at UCSF in the Gladstone. Me trying to like measure this in living cells, which was really interesting experience. Really, really beneficial. And I would like to thank Dr. Finkbeiner as well as Gabriella Novak and Vanessa Wheeler. So for letting me do this project, as well as the Huntington's Disease Society of America, or HDSA, for giving me a fellowship to do this research. And yes, that's part of why I'm like so interested in this topic. And I met a lot of really great advocates and affected families, and it was really, really an inspirational, but also very emotional experience. If anyone's interested more in Huntington's disease, I highly recommend you check out HD Buzz, and it's a really great blog and just community of people dedicated to sharing the science with the Huntington's disease community and pushing forward for a cure. So a little bit more about the biochemistry of glutamine. So if you've been following along, yesterday I talked about the amino acid asparagine. So glutamine has one more carbon, so it's a little longer. Its side chain's a little longer. This is the part that's going to stick off from the protein. And because it's a little longer, it's going to be more flexible. So it's going to allow regions of the protein that have a bunch of glutamine, such as an expanded glutamine tract, to kind of make more connections to other things. These both have an amide bond, and that's going to allow them to form these hydrogen bonds. So before I get too into the details, the key thing to keep in mind is just that, well, like charges attract, or opposite charges attract, like charges repel. And this is true of partial charges as well. And then in the case of these amide bonds, what you're going to have is you're going to have a partial pause. You're going to have partial charges. You're going to have these hydrogens be partly positive, these oxygens be partly negative, and voila, you get charge-based attractions. The reason why you have this partial charges is because, well, each of those different lines represents a bond, which involves the sharing of electrons. And so there's carbons and nitrogens and oxygens. These are all different atoms. Atoms are made up of smaller subatomic parts: protons, neutrons, and electrons. Protons are positively charged, electrons are negatively charged, and neutrons are neutral. If you have an even number of protons and electrons, you're going to have a neutral molecule overall. But if those electrons are distributed unevenly, then you can end up with the differential charges in different parts of the molecule. And this is what happens in the case of this amide bond. Basically, what happens is that oxygen and nitrogen, they're going to kind of hog the electrons. They're going to pull these electrons away from this hydrogen. It's going to leave the hydrogen partly positive, and it's going to make, like this oxygen is going to be partly negative, and therefore it's going to allow it to form these interactions. So we can get various types of partial charge interactions. The hydrogen bond is a special name what we give when we have basically a hydrogen attached to something electronegative. So when we talk about electronegative, that's something that's hogging electrons, so like an oxygen or a nitrogen. This is making the hydrogen partly positive. In a hydrogen bond, this is going to be, it's going to be complemented by an electronegative atom with a lone pair of electrons. So basically an unshared pair of electrons. So atoms can share pairs of electrons to form bonds, but they can also just kind of like have lone pairs, which they don't share with anyone. But those lone pairs are going to allow them to have a partial negative charge, and then they can share with the positive charge of a hydrogen. So you get this special kind of configuration, and you get what we call a hydrogen bond. This is going to be crucial for forming the backbone of proteins, forming things like alpha helices and beta strands, which can then like link up into beta sheets. And so this is all caused by these having amide bonds in the backbone of the protein. But in the case of glutamine and asparagine, you're going to have another one of these amide bonds in the side chain. And so these are going to be able to form additional interactions using the side chain of the protein, in addition to having those interactions in the backbone. And this is going to allow glutamine to form a bunch of different secondary structures, so like sheets and that sort of thing, with the backbone or with the other regions of a protein, or even another protein. If you have a lot of it, it's going to make it so that the protein can form other structures that the other proteins can't form. And so here was a recent paper that was actually looking at the shape of the kind of like aggregates that having a lot of these glutamines in a row can cause. And so they saw that having these longer repeat tracks led to like larger fibrils, so basically these larger aggregates. And you can see that you kind of have these parallel strands. And so you can see that the glutamine is kind of going to make it so that you can have these long connected strands, which are then going to be able to kind of pull other things into clumps and aggregates and things. So sometimes your cells can kind of try to try to handle all the aggregates through methods like using the ubiquitin proteome system. So basically putting this little protein called ubiquitin onto the ends of proteins that they want to degrade, then taking those proteins to the protein shredder in the cell called the proteasome, where those proteins can then get degraded. And so in the case of this Huntington's disease protein, however, if you have like too much of this faulty stuff, you can kind of use up the proteasome and kind of clog up the proteasome and not allow other faulty proteins to be degraded. So this is one way that Huntington's disease, that the protein is thought to cause problems. Also, those aggregates, they can kind of, because they can form those bonds with other proteins as well, they can kind of pull in the healthy versions of the Huntington protein, as well as other proteins. Research too, also like basically you can get these longer aggregates that clump up as well and are insoluble, as well as kind of smaller aggregates, like only a single copy of the protein, or maybe just even part of the protein, that are kind of soluble. And it's thought that these might actually play more of a bad role than some of the really big aggregates, where you have lots of these proteins, lots of mutant protein involved, might actually play a protective role by kind of hiding that protein from messing up other things. Whereas the smaller, more soluble products can cause more problems, because they're able to actually like go and interact with things. And so there's a lot of research being done on that as well. And the mRNA itself can sequester various things that are needed, as there's a lot of research trying to figure out the role of the RNA level. So a lot of the recent research has been trying to like lower the levels of the faulty messenger RNA, such as by using antisense oligonucleotides. So basically modified nucleotides, so like RNA or DNA, but modified so that it's chemically stronger, can be kind of used to block the using of this gene or of the mRNA. But the problem is it's been hard to target just the faulty version and not the healthy version as well. And there's been some toxicity problems. So there's been a lot of kind of like disappointing research or disappointing results from therapeutic trials lately, but there's still a lot of hope and more research going on. So that's the basics of Huntington's disease, and it's a really awful neurodegenerative disease, and I really hope that there are cures soon. But hopefully that helps you understand the genetic and biochemical basis of the disease.